Hello, Great Hope. Mr. Stolle here from your tutors. So today we're going to look into DNA replication and DNA profiling, okay? Now, what is DNA replication? So it is when a DNA makes identical copy of itself, okay? That's what you call DNA replication, okay? So when it makes identical copies of itself, but where does it occur? It occurs in the nucleus, because that's where we know that most of our DNA is found. So it occurs in the nucleus when during interphase before cell division. Then why? So that it ensures that we have enough DNA. The daughter cells of cell division have enough DNA. So enough DNA is available for daughter cells during cell division. Now, when we're talking of cell division, we remember that we're talking of mitosis, we are talking of what you call meiosis, okay? But in most cases here, it assists mitosis, because we know that daughter cells are identical. They need to have identical DNA, identical DNA to the mother and also identical DNA to each other, okay? Now, let's move on to the process itself. We need to understand DNA, the process of DNA replication. Now, we know that our DNA is double helix. So if it's double helix, meaning it's coiled, it needs to unwind first. So DNA double helix unwind, that's the first step. After unwinding, you find that your weak hydrogen bonds connecting the bases here breaks. So they do break and your DNA unzip. So this is unzip. As you can see, it looks like a zip, a zip of of your jackets and everything. So here we are saying DNA unzip. So don't forget to mention that weak hydrogen bond breaks and DNA unzip. That's your second step. Then the third step, we know that each of the strand is acting as a template to build the complementary strand. Then the fourth point, you must know that in building the complementary strand, we are using free nucleotides. These are free nucleotides that you are seeing here. These are free nucleotides build the complementary strand through specific base pairing. And we know that the specific base pairing, A can only pair with T, adenine can only pair with thymine, and C can only pair with G. Okay, so you need to specify that specific base pairing that we're talking about here is A matching with T and C matching with G, okay? Then as a result, we are forming two identical DNA molecules. One consists of new strand. So here we can see that we have an old strand, which is an original strand. Then the other one here is actually the new strand that is being formed, okay? So that's the process. Just remember to write these processes and you won't have a problem. Okay. Now let's have a question here. Name the process illustrated in the diagram above. This is DNA replication. DNA replication. Okay, why are we saying that? Because we can see that we have got DNA here, it's coiled here, and there is it's opening here, it's unzipped here, and also we can see that new strands are being formed. Complementary strands are being formed here. We can see the build up there of new strands. State the significance of the process. We can refer to the previous slide where we say we need enough DNA for available for cell division. Now, identify the parts labeled 1. So 1, what is 1? 1 is a deoxyribose sugar. Don't just say a sugar. Deoxyribose Okay, what about number 2? Number 2 is a phosphate group. Phosphate group. 3 is your nucleotide. Remember, when you see a nucleotide, remember here we have got the base, we have got the sugar, then we've got the phosphate group. Remember, a nucleotide is made up of those 3 things. Okay, so that's why we are circulating. A circle here shows that we are including everything is included there. What about number 4? Where is number 4 here? I can't see 4. I can't see 4. 4, here is 4. 4 is a nitrogenous base. Nitrogenous base. Okay, now, and we have got a nitrogenous base. And then we are done with that one. Then we can see we don't need to specify in this case because we don't know the actual base there. Describe how this process takes place. We have described in the previous slide, so you can actually remember that. You need to remember this because this is something that is being asked each and every year. Okay, now give one location of extra nuclear DNA. When you're saying extra nuclear DNA, it's outside the nucleus, outside nucleus. Okay, outside nucleus. So where do we find DNA outside the nucleus? It's actually the mitochondria. So the answer there is mitochondria. Okay, you can also talk of what is it, chloroplast. Because here they didn't specify whether it's plant or animal cell. Okay. Now let's move on to DNA profiling, where it's this is represented by bands. You can see this bands here shows the DNA profile, and the bands are of different, they are positioned on different position, and also they have different thickness. But you must remember that only identical twins have got identical what called DNA profile. But except all individuals except twins have unique DNA profile. So DNA profile is actually unique. But where do we use it? We use it to identify the crime suspects, okay, in forensic investigations, crime suspect, and also prove paternity, who the father is, and what called to the child. So paternity test, and also determine the probability or cause of genetic defects in the family, and also establish the compatibility of tissue types for organ transplant. So if you ever you want to do transplants, you need actually to test if actually there is a relation, what called there is compatibility there. Then identify relatives. So you use also DNA profile to identify relatives. What are the challenges? The challenges, people, remember, it's people who are in the laboratory doing these things, and people can make mistakes. So that's why we're saying human mistakes. DNA profile is expensive. So not everyone is able to actually to say, okay, I'm not sure if the child is mine, but sometimes you don't have enough money to pay for that what called the DNA or the paternity test. Now only a small piece of DNA is used, so we are not, it's not 100% accurate. So once a process is not 100% accurate, we cannot what called rely on it 100%. The method of profiling may be different in different laboratories, producing inconsistencies. Okay, then another thing also, DNA profile may reveal information about a person which could be used against them in a prejudicial way. So in this case, let's say someone, you show that you are HIV positive, or you have got some certain disabilities. In this case now, some insurance companies can deny to cover for you because they see that your profile is bad. Okay, that's why we are saying it's not good actually to show to have the profile published. Now, what about paternity tests? How do we apply now when we're answering exam questions? Because remember, in these questions that comes, you might be asked, who is the biological father of this child? So for example, in this case here, who is the biological father of this child? How now do you do this? first thing that you need to do here is to mark the bands that are matching with the mother. So here, as we can see here, the band that are matching with the mother is this one here because the mother have it, and also the other one is this one here because also the mother have it. So these are the bands that are only matching the mother and the child. Now, what do you do now? You now look on the remaining bands. The remaining bands should match the father. So you go here with your ruler. You can go there with the second, what called the second band of the child. You can see it's here. So you can see that it's matching the male 1 and male 2. Okay. Then we go down again and we see that the second band here that is not matching with the mother is matching with male 3, and we go down with our ruler. What's happening now? Okay, now go down with our ruler. We can see that this one is matching with the mother, then the remaining one is also matching with male 3. So as you can see that the remaining bands which are this one here, and this one here, they are matching with male 3. So that's the only way you can now say, oh, my answer to this is male 3. Okay, now give reason for your answer above. So when you're giving your reasons, you must always state that after matching DNA bands of the child with the mother. So after you match DNA bands of the child and the mother, all the remaining DNA bands in the child profile match that of male 3. But the question also can be asked, say, give reason why male 1 is not the father. Give reason why male 1 is not the father here. You can now say also, after matching DNA bands of the child with the mother, all remaining DNA bands in the child profile do not match. They don't match that of male 1. That's the reason. So that's this one is going to give you enough marks that will be allocated, or all the marks that will be allocated on that question. Okay, now explain precaution that should be taken when working with DNA samples in a laboratory. These questions are not common, but in this case here, if you are to have this kind of question, you must know that when you are dealing with samples, you must mark the samples clearly to make sure vials are not swapped. Okay, so to make sure that you don't mix up, and you must also wear gloves and a mask not to contaminate the samples with your own DNA. Okay, so you remember your DNA you can obtain it from your saliva, so we don't need actually to have your saliva DNA mixed with the sample. Use new and clean sterilized apparatus not to contaminate samples. So when you're dealing with this, you must always use a sterilized apparatus. Okay, now let's move on to the last part here, where we are asked to give the letters of the two people who are identical twins. So remember what I told you, you use the ruler to match the bands. So you match the bands there, and this is a question that you must answer. And give letters of the parent of person B. Again, you look at B and check two people that have their bands are matching with the child. Okay, then in that case you are able to know who the parents of B are. Now explain whether the collection of DNA from every citizen in South Africa to create DNA profile database for South Africa is a good idea or not. Okay, we are saying everyone, their DNA profile are recorded. Is it good? The first reason, you can say no. Why are we saying no? Because remember what we mentioned earlier, that DNA profile may reveal personal information about a person which could be used against them in prejudicial way. So in this case here, we are saying it's not a good way. Then, but again, also, it can also help us to find suspects quickly. Like, for example, here, if we say yes, it could be used to identify crime suspects, relatives, assist in organ transplant, and determine the causes of genetic defects, and or also prove parenthood. So this will help us to actually identify the crime suspects as quick as we can, and also also to identify who can actually assist with the organ transplant as quick as we can. Thank you so much. Until next time.